Good evening. My name is Keith Cole, and I serve as the Executive Director for the Wolf River Conservancy. I want to welcome you all here tonight for our lecture, which is part of our ongoing educational outreach to the community at large. Tonight, we're very excited about sharing with you a very special program on Copperhead Snakes presented by Dr. Maya Carrasco-Harris. First, I'd also like to acknowledge how we make all this possible. Our two presenting sponsors are Buckman, as well as the Crawford Howard Family Foundation. We'd also like to acknowledge all of our benefactors, which would include AutoZone, FedEx, Hyde Family Foundation, International Paper, Jim Karras Subaru, Ring Container Technologies, and Silvamo. Of course, all of our supporters, individual donors, volunteers, and corporations are all important to us and help us to deliver on our mission, which is to preserve and enhance the Wolf River and its watershed as a sustainable natural resource. Some housekeeping details: we ask you not to record tonight's program. We are recording it for you. If you registered for tonight's program, you will receive a link in the next few days that will allow you to look at the recorded program at your own time and convenience. If you have questions during the program, please use the Q&A feature and not the chat feature to register your questions. Our Education Director, Kathy Justice, will be monitoring the Q&As or the questions, and she'll present those to our speaker at the end of the evening. Now, it's my pleasure to present to you our speaker for this evening, Dr. Maya Carrasco-Harris. Maya will discuss the ecology and behavior of copperhead snakes in Overton Park. This venomous pit viper is a common feature of eastern forests and woodlands. Due to its subtle behavior and camouflaging pattern, this frequently underdetected snake has generated significant fear and persecution. The antidote to fear, we believe, is knowledge. And so tonight you will hear from Maya information that will help you look at this native animal in a new light. Maya moved to Memphis to pursue her first job as a high school biology teacher, and she and her dog frequently sought solace on the trails of the old forest of Overton Park, like many of you out there tonight have done as well. She started wondering about the challenges animals face living in urban environments and pursued those questions by studying the spatial ecology and genetics of copperheads, particularly in Overton Park. She received a PhD in biology from the University of Memphis in 2020. She joined the Overton Park Conservancy as a programs manager to develop experiences that promote curiosity, whole health, and self-expression in the heart of Memphis. And we're glad she's with us tonight. Please help us welcome to our virtual stage, Dr. Maya Carrasco-Harris. Welcome.
Thank you. Well, thank you for the introduction, Keith, and Kathy Justice. Thank you so much for the invitation. I appreciate it so much. I'm very happy to be here. Most of this work, all of this work actually is going to be from my time at the University of Memphis. I'm currently the programs manager at Overton Park Conservancy, so I'm very happy to get to talk to you about research that I did in Overton while I was a graduate student. And I want to go ahead and acknowledge that that work was funded at the time by the University of Memphis, the National Sciences Foundation, and the Memphis Zoo. So with that, I want you to take a look at this picture that I took while in the field and see if you can spot the copperhead. So if you're wondering where it is, it's just northeast of center. And I think that this is a really good reason for people to stay on the trails, whether they're at Overton Park or they're along one of the Wolf River greenways. Just make sure that you're staying on the trail because these animals are very adapted to that leaf litter in these environments. So like Keith mentioned, my story begins here on the trails of Overton Park. I had an insanely crazy dog at the time, and she needed to go out all the time. And I was kind of losing my mind in the classroom, so I needed to go out all the time. And while I was out there biking, running, hiking, I just started wondering, what effect am I having on the animals living here? I figured that there had to be something. I had an ecological background prior to becoming a teacher, and so that I knew that there was some type of effect. I just didn't know what that effect was. And then kind of in the bigger sense, I had just moved to Memphis, which is the largest city I've ever lived in. And I just constantly thought to myself, how does this urbanization, this suburbanization, affect animals kind of in a grander scale? And that wasn't an irrelevant question to be having at the time, actually, because more people today live in urban areas than in rural areas. In this map here, the darker the shade of blue is, the higher the percentage of the population of that country living in an urban area. And by 2050, that map is going to look like this. The United Nations predicts that by that year, 66% of people will be living in urban areas. So as more people move to urban areas, they bring with them the desire to still seek the outdoors, but also this conflict that occurs just from the nature of urbanization, which can lead to biotic invasions. We subsidize wildlife. We bring in non-native species that often are not just plants, but often have the insects associated with those. Those affect ecosystems. Because of the density of us and the animals around us, we end up spreading disease more likely. We change land cover, whether that's making trails, making roads. We add impervious surfaces that change the landscape around there. And then we also create fragments. Overton Park is a fragment of a natural setting within an urban area, and that changes the ecosystem dynamics. And then also, we tend to modify even our park landscapes. We prefer parks that look like the one on the right to the one to the left that are often more simple. They're easier to manage. You can mow them. You can see what's around you easily. We take care of water by creating drainage systems so that our cities don't flood. But going from a complex to a simplified ecosystem has effects. And often in biology, we think about the more complexity an ecosystem has in terms of spatial diversity, the better it is for the biodiversity. So moving a little bit closer about the consequences of urbanization, they affect all taxa, but some taxa are more studied than others. For example, birds are a lot easier to study, and I also use eBird and collect a lot of birding data on that app. And so because of their easiness to spot, to see, to hear their calls and their mobility, avian research has been very prominent in urban studies. But what has been less prominent are studies on non-avian species. And so McKinney did a review paper and found that in non-avian species, you're still seeing changes in species richness, which means that there's just less types of animals in more urban environments. And that is something that you can see just by going to our different greenways. You can go in your backyard and count a few different types of birds or animals. But if you visit one of the Wolf River Greenway areas, or you visit Overton Park or Shelby Farms, you're going to encounter different kinds of species. So that species richness leads to also reduced biodiversity. Globally, we have more forest fragments than whole fragments. So Haddad and his colleagues looked at forest cover and found that 70% of the remaining forests globally are within a kilometer of the forest edge. And forest dynamics change when they're fragments. And so the effects of that was a reduction in biodiversity between 13 and 75%. So this is impairing the ecosystem's function. And so clearly what we should be thinking of is, how do we then manage fragments as we are creating them in our environment? So zooming in a little bit more closely as in terms of behavioral ecology, when we think about globally, there's a decrease in biodiversity, but what is left there doesn't have the same behavior necessarily that it had in its non-urban environments. Some of those behaviors are changes in boldness. I took this screenshot off of a news site just a couple of months ago where Hank the Tank had invaded 21 homes, and this is a large bear. He learned that there were anthropogenic food sources that are very high in calories, and we have the same issue with the squirrels in Overton, like this one that's taken a piece of pizza out of the trash can. And so animals will exhibit more bold behavior in urban areas. Typically, the ones that stay might be a little bit more bold, and they can handle the disturbances that humans bring with them. But their populations might also be constrained to certain areas of cities, for example. So Chicago coyotes tend to have home ranges where they try to avoid humans, and they also change their behavior to be more nocturnal so they can avoid when humans are out most. And there's also been studies showing that hawks tend to have smaller home ranges where they're spending most of their time, just because they can see prey easier because we've made very simplified ecosystems for them. But again, the question that I then kept having was, well, what about urban reptiles? Reptiles are unique in that they are not as mobile as many mammals. They're not as able to move from an area as the birds are. And then certain man-made structures really present issues for their mobility. For example, railroad crossings, roads, those could be difficult, if not impossible, for animals to get across. And so when animals cannot get across structures that we see as very simple to cross, what ends up happening is that they're not able to get up to other populations, and you have reduced genetic flow, which limits the genetic diversity, which puts animals that are relatively close to each other in danger of inbreeding in the future. So my study is particularly looking at urban refuges, because as we continue urbanizing, we are more and more wanting to create parks so that people in these urban areas have an easy way to access natural areas. So you've got many different kinds of natural fragments that try to replicate kind of old forest conditions, such as the one in Portland, one in Atlanta, New York City. We visited an old growth forest in Indiana recently, and then of course Overton Park. And these are a little bit different than regular city-run parks in that they have complex ecosystems. They're not devoid of the same types of issues. For example, they have more edges that are often roads, and so animals that are in these refuges might not be able to leave. They tend to have more biotic invasion, so non-native species that create changes in the physical habitat in the plant matter or the animal matter that's there. You also have, these parks also have to deal with urban temperature changes. Urban areas tend to be a lot warmer than natural settings because of all of our impervious surfaces that reflect light and heat. They have to deal with pollution. Of course, there's recreational use and sometimes management that's also occurring. And so not all animals that would be in natural areas can survive in urban natural fragments, even if they're there. So if you're not familiar with Overton Park, we are located in the heart of Memphis. There's about 342 acres in the park, and 126 of those are what we consider old growth forests. They're not managed like the rest of the parkland is. It is bordered by some large thoroughfares. You have North Parkway on the north side, East Parkway, Poplar, and then McLean. So some very busy roads. So if you're an animal, it would be very difficult to cross those, whereas it's not as difficult to come into Overton if you're a migrating bird. We have a lot of amenities to offer, from dog parks, playgrounds for kids, open green space, to trails, which is really where the study focuses. Those that trail network in the woods is what allows all of these different species to still cohabitate in this space. We have different types of snakes, turtles, skink populations in there. And so then even more specifically, I studied the copperhead snake because in a previous study, it had seemed to be the most prolific snake, which is what I found to be the truth. They're also relatively easy to work with as long as you isolate the head. So Copperhead Dicystrodon contortrix is a venomous pit viper. I've shown two different pictures here. One of them is a little bit darker than the one on the right, is a little bit lighter and brown. So they can vary from pink to grayish brown. It used to be thought those were different subspecies, but now they're all characterized as one species that has a very large geographic range. They get to be about 20 to 37 inches long. So if you ever hear someone tell you that they killed a 4-foot copperhead, they did not. They are what I like to call stout, kind of chubby snakes. They're not super great climbers, but they're an ambush predator. So they do what you see in both of these pictures. They coil up, they sit, and they often wait for prey to go by. Sometimes they do take on a more active role in their hunting. They're generalists. They'll eat insects when they're smaller, amphibians, small mammals, whatever they can actually get a hold on. Cicadas are a favorite when those are out. They have those iconic bands on them. I like to think of them as Hershey's Kisses. Some field guides call them saddles, but they're unique to each snake. So every snake that I caught, I got to marvel at the fact that it had different banding, just like a zebra does, like your fingerprint's different than other people's. And they have about 13 to 15 of those bands. Sometimes they meet symmetrically in the middle. There were other times when the snake, I would maybe call them ugly, but they didn't have bands that met symmetrically. Or sometimes, interestingly, it looked like the Hershey's Kisses had melted down the side of the snake. So there's incredible variation in these beautiful animals. And of course, they do have that reddish head, which is where they get the name Copperhead from. Because they can blend in so well for their environment, they tend to do fine in suburban areas, and as we discovered in Overton Park, because they can remain cryptic. They have keeled scales. If you were to find maybe a rat snake in your backyard and you look at its scales, they're smooth, but the keel is a little ridge on the scale that gives it a more matte appearance. And then importantly, they also have these pits on their face, which is right between the nostril and the eyes. And this is for heat sensing. So they can detect prey by heat like other pit vipers do, like rattlesnakes. This is particularly important for them in the summer when they become more nocturnal. It allows that heat differential to become a little bit greater, detect animals and help them thermoregulate. One fun fact that many people don't know is that they actually give live birth to about 2 to 18 animals. I never saw clutches that large, but they'll stay with their offspring for the first couple of weeks until the first shed. And actually pit vipers, so the family that includes rattlesnakes, tend to be a little bit more social of the snakes. And sometimes in rattlesnake species, we've seen that they will often assign another female to kind of guard all of the young from predators, which is kind of cool because we often think of snakes as loner species. When they're born, this picture, you can kind of see it, the juveniles will have a little green tip, and that's for caudal luring. So they can sit in one spot and then wave that tail around and hopefully attract some prey to eat. Another interesting fact about breeding is that, especially here in the southern range, they have 2 breeding seasons. So the most likely time you're going to see the copperhead snake is either in the spring, particularly when they're moving from their winter site to wherever they're going to find a mate, and then in the fall again, where they'll mate one more time and then go and hibernate. If a female mates in the spring, she can use that sperm to then produce offspring for that summer. But if she mates in the fall, she can hold that sperm until the next season and then give birth. So here's a map just showing a little bit of their distribution. You have a different copperhead species in Texas, but everything pretty much east of Texas is now considered one species. And one thing that this map kind of highlights is because of their broad distribution all the way up to like Connecticut and Massachusetts, they are kind of can serve as a canary in the coal mine. They're not of conservation risk. They're generalist eaters, and they tend to be cryptic and hide very well, so they can survive in habitats that other animals can't. And so they could, it's been suggested they could be a good management indicator for different natural areas. Do you have copperheads or not? Similar to how you would look for other key species. So I talked a little bit about Overton Park, which is surrounded by impervious surfaces, development. And then my other study site that I was working at was the Meeman Biological Field Station. If you're familiar with Meeman Shelby State Park, this is close to that, just a little bit northeast of there. And that site is run by the University of Memphis. It does not have recreational trails, and the only people that really use the area are other scientists. And so you have a lot less human disturbance in that area than you do in Overton Park. And so this study is really, because we have just 2 sites, there's not any more urban sites, there's not any more rural sites, it's really a good case study. But everything that I discussed today, there's just opportunity for more research and more diverse urban habitats. So I'll talk about mostly spatial ecology and then a little bit about genetics for the questions that I answered in my dissertation research. So of course, studying copperheads means we had to catch them, and you do that safely because copperhead snakes, of course, are venomous. So we did this with hook and tongs primarily, and then we also set up funnel box traps, and so that's what you see on the right. So you install a drift fence, just like chicken wire fencing, And then if an animal comes upon it, it either has to make a right or it has to make a left. And when it does so, we'll have funnel box traps on either end of that fence. Animal goes into the funnel and drops into a box. And you hope that it can't find its way back through the funnel. If you happen to leave a snake like I did once and think you're going to come back in half an hour for it, it can just prove that they can find their way out. Once animals were captured, we took them to the Memphis Zoo to process them. So I got a lot of help from the curator, Chris Baker there. I've worked with mammals before, and copperheads are a little bit easier. I only have to worry about one point of contention there, the head. And so we isolated them in tubes safely. We sex the animals by using a probe. If it's a male, the probe should go in deeper onto one side of the tail because they have two hemipenes. If it's a female, the probe will not insert. We then gave each animal a tag. This is similar to the microchip that you gave your dog. I was able to read that later to see if I had recaptured an animal. We digitally photographed the animal so that we could measure them on the computer, measure their snout vent length. So snakes, surprise, they do have a body and a distinct tail. So the vent is where the body ends and the tail begins. And so that's what I'm pointing to with the hook there. Animals that were large enough, we then wanted to track for a year. And so we were implanting radio transmitter tags to them. So those are different than the little microchips, which are passive. The radio tags actually emit a signal. So to do this, snakes underwent a surgery at the Memphis Zoo, and they were very lucky that they were in the fine hands of Dr. Felicia Knightley there at the veterinary clinic. We put them to sleep by putting them under anesthesia. Sometimes I think they were smarter than we were, and they would just stop breathing. And so it was difficult to put some of them to sleep. Then to keep them to sleep, we intubated them, which is what you see here on the right. A snake's trachea is actually all the way at the front of its mouth so that they can move it out of the way when they're eating a larger meal. So intubating them is pretty easy once you have them asleep, which can be difficult to tell because snakes don't have eyelids. And so even though the snake's eyes are open and it looks like he's staring at you, he's actually under sedation. We would undergo cleaning procedures before the snakes had transmitters implanted. And then here's an example of one of the transmitters implanted. So part of the body of the transmitter is a battery, and then the other part of it is the radio signal that's being emitted. And part of the reason why many of the studies that have been done in urban areas and in other areas were on mammals and birds is it's just easier to put radio collars on mammals. Everybody's seen that on Animal Planet before, or to put a backpack on a bird. But with snakes, it's a little harder. They don't have real necks, so you can't put a collar on them. And so technology is really what stopped more reptile research from happening at the same pace that you had bird and mammalian urban research happening. And so being able to get transmitters of this size then allows you to study some of these smaller, more difficult to find animals. Once a snake had its tag implanted, we would release it into the forest and then go and re-find it several times a week. I had an army of about 16 undergraduates throughout the few years that we did the study that helped with that. And so you can see Vaden to the left there is tuning into the radio station of a particular animal, and then a beep is being emitted every few seconds, and you use that to find the animal. The louder the beep is, the closer you are to the animal. Once they found the animal, we would collect a lot of data. We were very thankful that we were even able to do this research. And so once we were there, we collected all kinds of environmental data, behavior of the animal, and then of course, GPS coordinates to get the location and the time. So let's start off with some of the differences that we saw between Overton and Neiman. The first big question is, how do copperheads living in Overton differ in their morphology? We were taking that data in the lab, and then in their movement behavior, which we collected out in the field, from those that were at Neiman. So just real quick, some of the big differences you saw were in size. Neiman snakes were on average almost 2.5 times larger than Overton Park snakes. And this wasn't a surprise. I had people that were actually real herpetologists who would tell me that they thought the snakes were smaller, and even just our regular park users who walk through there and see the snakes crossing the road during the spring migration would suggest that they looked like they were on the small side. And so we confirmed that with data, finding that on average, the copperhead snakes in Overton weigh about 4 ounces. The units that you see on the left in the graph are the units that we use in science in grams. So I wanted to translate those so you could kind of understand what that looks like. In terms of length, same story there. The Meeman snakes tended to be larger than the Overton Park snakes. Average in Overton for males was about 20 inches. Average in Meeman was about 24. There was a pretty large range. The largest snake that I saw in Meeman was about 1.7 pounds and 42 inches. So that was a large animal, but that was an outlier. So to give you kind of some understanding of what does that look like, Chris is holding an animal that's more representative of maybe a larger Meeman snake, and Janice on the bottom has an animal in a tube that's representative more of an Overton Park snake. So if you see a large snake in Overton, it's more likely you see a rat snake than you see a copperhead, because they're just not as big as we think they are. And so the big question there was answered. Overton Park snakes are smaller than their Meeman counterparts, but they're similar in body condition. So that's another metric, kind of like your BMI that the doctor might take for you. They're not just really skinny, long snakes. They're about the right size. And there's a lot of explanations for this. In this study, we can't say why that's the case. There could be differences in diet between Overton Park snakes and Meeman snakes. We didn't do stomach analyses from the snakes to see what they were eating. There could be growth rate differences. Reptiles are indeterminate growers. So if they eat a lot initially, they can grow faster than one that doesn't have as much food availability initially. And so you cannot age an animal based on its size. And so that it could be another reason, is perhaps there's skewed population demographics. I found a lot of snakes in Overton, but a lot of them were smaller. So it could be that you have a young population that tends to die quicker. That's one potential option. And there's also the option of genetic differences. These are two distinct populations, and one population might just remain smaller than the other. So with our study, we really couldn't detect that. So the other question that we were trying to answer in the field was, how do their home ranges differ? So we talked a little bit about how bird home ranges might be smaller in urban areas, for example, hawks, because they can find food easier. And some of the coyotes also will modulate their home ranges to avoid humans. And a home range, I like to explain to people, is wherever you go, for example, in a week is your home range. So my home range would include my house, Overton Park, the office, my friends' houses that I go to on a regular occasion. So it's just where you spend your life. For animals, where are they mating? Where are they rearing their young? Where are they finding food? And then sometimes you also consider, where do they hibernate? There's a couple of ways that we like to talk about home ranges mathematically, but there's two models that I'll present are here. So this green section is the forest in Overton Park. They've got two different maps on here. And each of those polygons that you see, for example, on the left-hand side, is a home range for one animal. And so the top northwest corner has a yellow polygon on one side. That's one model, shows its home range like that. And that's essentially, if you were to put pins up on a board and then put up a rubber band around those pins, everywhere that animal had been, we considered it its home range. The model on the right-hand side is a more mathematical probabilistic model, where you're trying to find areas of high use compared to areas of low use. And so we used many different models to kind of quantify home range. But at the end of the day, what we found was that home ranges in Overton were Meemans were about 6 times bigger than those in Overton, which they could have fit. The home range sizes of the snakes in Meeman, just to compare here, could have fit in Overton. But they just weren't using that space the way that Meeman snakes were using that. So their home ranges were a lot smaller. And so since we only had one urban site and one rural site, we went to the literature to compare, well, what is the average home range size look like? Are the Meeman ones even normal home range sizes for copperheads? And so I compared it to copperheads in Nebraska that were wild, and Connecticut, and in Alabama. So very different geographic areas. And what we found was that the Meeman home ranges were normal, but the Overton, for the most part, were smaller than all of those other rural sites. So we know that Overton Park snakes have smaller home ranges than the Meeman ones and other rural conspecifics. And again, that could be because of multiple reasons that we cannot determine from this study. There could be denser food availability, so the snakes don't have to move as much. There's different types of species of snakes in Overton than in Meeman. There's more biodiversity in Meeman, so they might have not only predator release, but also competitor release. They're not having to compete with other types of snakes, so they might not need to move around as much because of that. There's also human disturbance. So is there the potential that humans using the trails cause the snakes to move less because there are these barriers within the woods? And it could be a combination of all of those circumstances. What we do know from some other pit viper studies that aren't necessarily done in urban areas, but are done in areas that have some disturbance, is that it just depends on the type of animal. Some of the rattlesnakes that were studied can adjust to the human disturbance by remaining concealed more frequently, so changing their behavior so that we don't see them. Some of them do decrease their home ranges, so this wasn't an unexpected finding as well. Some species do not change their home ranges at all, so it really can depend on the species and their sensitivity to human disturbance. So because we talked about human disturbance, we can focus then on what about the trails in Overton Park? Are there differences in snake movement in relation to the trails? The trails are really important for human recreation in Midtown and Memphis, but we do know from previous studies that animals respond to what we consider low impact use, even if it's just hiking. And the distances that people think they're giving animals are often not as large as they think that they really are giving them, and have a greater effect than they know. So we took all of the points that we had collected for every single animal, which is shown here as black dots. And we measured the distance to the closest trail, which is represented, that distance is represented by the pink line. And so then we created models to see, does the distance this animal is to the trail then affect how much it's moving? And I would have thought, yes. Animals that are close to trails definitely are moving less than animals that are further than trails. But that's just not what we found. We found that they move differently based on whether they were male and female, which others had already recorded that. We already know that males move more, and we already know that copperheads move more during the breeding season when they're going from wherever they overwintered to wherever they're going to find mates and spend the rest of the summer. And so we were really surprised that they didn't really change their behavior based on the trails. Eastern Massasauga rattlesnakes did show that they altered their behavior around human exposure, so we thought there was some potential there, whereas timber rattlesnakes did not alter their movement when there was logging behavior. So again, it can just depend on the animal species as well as the individual. In our modeling that we were considering, the greatest factor of whether or not an animal was moving was really just the individual animal itself. That was some animals move more than other animals because they choose to. So another way that we wanted to look at this was, are animals kind of choosing to be around trails or choosing to avoid trails? So what we did with that to study that was we take an animal's movements. So all of the black dots here represent every location that I found Hootie, which was one of my very first animals pictured here. We then used computer simulation to pick up all of those movements and just move them all around the park randomly. And then we compared, are animals closer to trails than you'd expect or further from trails than you would expect compared to those random distributions? And what we found was so messy. In science, you like to see a graph that goes in one direction and is significant in that direction. But what we found that's shown here in this graph is that that some animals were further away from trails than you would expect at random, and some animals were closer to trails than you would expect at random. There was no clear pattern in one direction. So again, the individual really mattered here. And when we plot who's significant and who's not significantly different, the ones that tended to be further away from the trails kind of ended up in the part of the park that doesn't have a lot of trails because of a fence that divides Overton Park from the zoo. And then the ones that tended to be closer to trails are in this southern section of the park in orange, that tend to be where there's a lot of volunteer trails and you've got some more roads. So the question then is, well, are the animals choosing to be closer to trails, or do they, if they're less tolerant, then they move to areas that have less trails? And we just don't know that because the study isn't designed to follow copperheads from birth on. And so what we can just say is that wherever they are, is going to dictate their distance to the trail, which is kind of one of those, well, no, duh, of course. We just don't know whether or not they're choosing to be there or if they can pick up and leave if they feel like this area is too busy for them. So overall, what we could say is copperheads are not altering their movement behavior based on the distance to the trails, which is comforting as somebody who, is working in a park and wondering if we're doing the right thing for the animals here. Kind of what we land on here is that their behavior, they do not become defensive when you come by, and their crypsis, their ability to blend in really well, hopefully you've already found the copperhead in this picture, allows them to remain tolerant to the relatively low level of disturbance that human hikers present in the forest. A similar study that kind of hits on the same vein actually found copperheads close to roadsides and compared their hormonal stress response to copperheads that were found further away from the road, and found that those copperheads that were closer to the road just had decreased stress response. They get acclimated to the stressors, similar to the way that we can get acclimated to living on a noisy street over a period of time. A similar species, the cottonmouth, is also found to kind of have relatively low levels of stress response to human disturbance. And other animals that act in cryptic ways, such as the eastern Massasauga rattlesnake, sometimes they just depend on that behavior. Although it's a rattlesnake that's also venomous, in trials done, 60% of the animals would not rattle when there was human disturbance, similar to what hikers do, like maybe stepping over a rattlesnake. And that behavior was consistent over time. So if an animal didn't rattle when a hiker came close to it, it was unlikely to rattle again the next time a hiker came close to it. So these animals are really just adjusting to our presence by maintaining kind of a cool, calm demeanor, in particular the copperhead. But I just hope that this serves as a friendly reminder to practice your leave no trace principles and stay on the trails. And if you're somewhere where a copperhead happens to be crossing the trail, give the animal time to cross the trail. If it's not moving and it's sunbathing, then just turn around or stay and admire for a little while. I don't recommend trying to walk over the animal, as that might feel like a threat to the animal. My next question that was related to field work was, what are the habitat features of Overton Park copperheads? We have a different kind of forest. There's a lot of non-native species in there that provide a lot of cover. So what I was hoping in the idealized situation to do was to collect habitat variables. I would find a snake. I would capture all of its relocation data. I would go and look for it several times each week for a year, determine what are the areas it likes, what are the areas it doesn't like as much, and then capture all of this habitat data at each single point to then be able to create a mathematical model. That was the ideal solution. But in reality, the forest is a messy place, and ecological data is hard to collect and even understand. There is a snake in the middle of this quadrant. And you could see here the difficulties it would be to collect leaf litter, at what part you could collect leaf litter, what is the amount of vegetation over this, how do you quantify all of these variables and put yourself in the mind of a relatively small animal to understand what bit of this environment does it like. So after all this data collection and hammering it out, trying to create a mathematical model that made sense, that was useful, that was predictive, all we could find was kind of the things that farmers already know. They like forest canopy cover, and they like logs, which anybody that has a wood pile and lives out in Eads knows that these two are some important habitat qualities. But those were such limited proportions of what we know the snakes are choosing in their environment. But it was a good exercise because no type of modeling had been done like this before with an animal like this. Most of the time when we do habitat modeling in ecology, it's with large animals like elephants, where those habitat variables are a little bit more obvious. And so this was an exploration that really no one had attempted to do before. And we found some things along the way that would be interesting to try differently for future studies with small animals. The last thing was I took samples from animals to then compare their genetic diversity, because again, their spatial ecology is limited. There have been roads bordering the park for at least 100 years. Previous to that, there was farmland. And so we believe that there is a bottleneck at some point where the animals couldn't leave the park any longer because the traffic is too high. And so we're not exactly sure when that period was, but as you imagine, East Memphis continuing to grow, there came a time when these animals could no longer access other green spaces. So we asked the question then, well, how do their genetics differ? This was really easy to answer with copperheads because another cool fact about them is they have nucleated blood cells. So you and I have red blood cells that don't have nuclei, so that there's a little cup in your red blood cell instead of a nucleus so that you can better hold oxygen. Snakes don't have that. They have a nucleus within every single blood cell. So you can collect a small amount of blood from them and then get a lot of clean cells to then do genetic studies with. So we looked at a couple of measures of genetic diversity. First, how many different types of alleles do you have for genes? So you have a gene, and then there maybe are several different types of that gene that you could have. And we found that the Overton Park snakes had fewer alleles than the Mimim snake. So there was less types of copies of genes than in the other site. And so kind of following along with that, they had less genetic diversity. So you would think that with those two pieces of data, that this population is isolated from other copperhead snakes, then there must be inbreeding. There's lower genetic diversity, there must be inbreeding. But we didn't find that in this population. Their inbreeding coefficients were pretty similar, so there are no we don't believe they're currently inbreeding. And some studies estimate you have to get to a population less than 40 for animals to actually be inbreeding. And I caught over 200 copperheads in my time in Overton over 3 years. And so the population is sufficient there. And it's also really been blocked for just a relatively short amount of time compared to the larger Western Tennessee history. 100 years is not that long. And there might have been years even closer to that where they could have crossed and got over a less busy road and gone over to farmland on the east side of the park. They were genetically distinct populations from based on locality. So if you were to hand me a vial from one of these two and say, can you tell me if this blood sample's from Mimim or Overton, you'd be able to do that because these snakes are distinct. There was one animal that his signature almost looked like he was an Overton Park, or he was a snake in Mimim that almost looked like an Overton Park snake. And it really made me wonder if somebody captured an animal and did their own translocation experiment there. So what we kind of know then is that there's lower genetic diversity, which is expected, but that they're not inbreeding now. And so that might take a few more generations. At this point, you might have only had 5 to 10 generations of copperheads that have been isolated. And so we might re-ask the question 10, 100, 200 years from now, what's going on with that population? And that's really important to kind of think about that time scale when you're thinking about management and natural fragments, because typically we want to rush in and see everything, how is it doing right now? But for animals, it can take a really long time to understand what the effects are because it takes so long for those populations to shift and to change. And so kind of the overall message is that the copperheads, snakes in Overton Park are currently adapting to the recreational use, and they're so far at least genetically resilient. And so I think this is it's a good thing for us, and it should encourage us to continue protecting habitat that is complex, that would provide a space and a niche for a wide variety of taxa. Even if they might be taxa that we find scary today, they're still valuable in their environment. I have examples of a couple of other predators here, including a fox and a coyote. So again, all of this research was conducted while I was at the University of Memphis in the dissertation program, and I would be remiss to not look back and say thank you to all of the undergraduate assistants, the people at the Memphis Zoo that helped. I had a fantastic committee that was also very helpful for me, and several people from different departments even that helped me understand my data better and understand what was going on. And then specifically, I want to just point out some of the co-authors we had here, including Dr. Cole and Dr. Reichling, Dr. Bowman, and Dr. Kwan, Mendel, and Siniscalchi. Those were all very helpful, and these papers, the study wouldn't have been complete without any of those people. And so with that, I like to open it up for the Q&A.
Okay, thank you, Maya. That was really good. And I'm hoping my video camera will come on pretty soon. So yes, thanks so much. It was very interesting. We have one question, which is which you can answer. Do all vipers give live birth?
I believe vipers do give live birth. That I think the Latin word viperity is hinting towards that live birth.
Mm-hmm.
Don't hold me to that.
And then just in case there are some people who don't know that all snakes are not vipers, right? So
correct.
Right. Mm. So some snakes do lay eggs, but not vipers. Okay.
Oh. Yeah.
Yeah. So I guess I was really curious about the future of this population and whether it's growing. It's growing less genetically diverse over time, but yeah, I guess I'm wondering what the future holds for the Overton Park population, and if there's any possibility of connecting it up to other habitat.
Yeah, that's a great question. I have kind of wondered myself, what if there was a greenway that continued to connect? The Shelby Farms green line doesn't end so far away. It ends right there at Tillman. And I think that there are projects to continue that. I don't think that they'll hook up with the park. And then if they did, would that greenway be sufficient habitat for an animal to move around? I know people have seen snakes on the green line. I've seen snakes on the green line. I've never seen a copperhead on the green line, but I'm usually moving pretty fast on a bike. And so I wouldn't be able to see them if I did, but I've never seen one walking or running either. So I'm not sure if it would be the right type of habitat, given it's so long for any animals to actually move. And somebody asked me recently, they thought they had seen a speckled king snake, for example. King snakes are top predators. They can eat other snakes as well as other animals. And in areas where there are no king snakes, your population of copperheads tends to be higher. And so that, I think, could be one of the reasons why the population seems so dense in Overton. They don't have these other predator snake species. And so when someone asked me, well, should we just bring in king snakes to Overton Park? And that's a tricky question. I mean, you could drop off an animal, but they're territorial. And so how many could you actually drop off before they start to remove each other from the habitat? Yeah. And if you bring them in, you're also bringing in potentially other diseases. You're maybe constantly always having to do management work. And so those are good questions to understand. Should we bring in other animals? And the same goes for the copperheads. Should we translocate copperheads, do some population switching to then provide a greater genetic base for these animals? I think there are very few Midtowners who would agree to that plan. But those are the kinds of questions that people who go into the field of conservation management, they want to know that data so that they can then create action plans. If this animal was one of those of concern species, yeah, maybe. But given that they are of least concern in terms of IUCN ratings, it's not on anybody's agenda right now to be translocating copperhead snakes.
Right, we've got plenty. So yeah. Okay, more questions have come in. The same question: have you ever been bitten during a capture or during your study?
I get that question every time. I should have just addressed it. I was never bitten. I think that with my loose data, the demographics of people that get bitten are males in between the ages of 8 and 24, and the famous last words are "watch this." And so I was fortunate to be very safe with the animals, and I'll be completely honest, very few strike attempts even. Often I would pick them up with tongs, and their defense is freeze behavior. So I have stepped on animals, done a complete 180 on one time. I felt awful, and the animal, it just sat there. But that's not the case for everybody. We did have a runner on a trail this spring who moved off the trail to let a dog and a human pass, and she must have stepped right on one, and it bit her. And so she was hospitalized for that. They're not deadly. And so unless you're very young or immunocompromised or very, very old, a copperhead bite should receive medical attention, but is unlikely to too serious, you might get a secondary infection, which could be often worse than the actual venom bite. And so most of the animals that I caught were pretty chill. And
Sweetie
you know,
pies, yeah.
not all of them. The hotter it is, you know,
Yeah.
the more ferocious they can be. But we were always very careful. I had 4-foot tongs and hooks, so I kept them a distance.
Yeah. Here's a good question. Do snakes brumate rather than hibernate?
I would say that they don't do a true hibernation, because in January, when it would get a little bit warmer, you know, we have those beautiful days when everybody's in the park. I would go out and I would find some of the animals out basking. So if an animal feels sick, they will try to just elevate their body temperature by basking. And some of the issues with that, with some of the snakes and that I've heard about, not any that I saw, but if a snake also then has a meal and then has to go back down and go into kind of a torpor where their metabolism is low, they can actually die just from the food rotting, because they need their metabolism to be elevated in order to digest the food. And so if there's a warm couple of days in January and the snake comes out, and then you go right back to winter weather afterward, if the snake had a meal, that can be problematic. So that's one of those climate change effects that land managers are actually looking at for species of greater concern. Is, oh wait, our animals they're getting confused by these differences in unseasonal, uncharacteristic weather.
Yeah, there's a lot to worry about.
Yeah.
Let's see, this goes back to the biting thing. Are records kept on the number of humans bitten by copperheads in Overton Park?
Probably not in Overton Park. When I found data on bites, it was provided by, I think, health organizations.
Mm-hmm.
So we just keep them, you know, if we find out about them, you know, we discuss amongst ourselves, like, oh, can we learn more? You know, what happened, but often that person's not connected to us. It's only if they are connected to us that we usually hear about it.
Right, and you probably don't hear about it often, it sounds like.
Right. I think probably more dogs get bitten. You know, we do ask that everyone keep their dog on leash. The Old Forest is a state natural area, and it's one of the few that's in an urban area. And so footprints matter to the plant life. And then also the behavior of your animals matters, especially even just sense, you know, dogs going off trails and spreading their scent, not even just stepping on animals, but spreading their scent can change the behavior of other animals. And so we just ask that people keep their dogs on leash so that their dogs aren't getting bitten and their dogs aren't biting others.
Yeah. I work at the greenhouse over in the 13 acres at the East Parkway compound. I have seen one copperhead in the greenhouse by my compost pile. I've also seen decays and cottonmouth snakes. It's not a question, that's just a report.
All right, I would be very interested if you did see a cottonmouth snake. They look similar to copperheads when they're younger, but I think after the channelization of Lick Creek in the 30s, we don't really have the right type of environment to support them. So I have wondered, what would it be like if we removed the channelization and made that a natural waterway? Of course, everything north of there would flood, so roads would be very unhappy. But maybe we could get cottonmouths in that forest. So I'd be really curious if there had been one, and I'd also love to see, you know, a speckled king snake out there, because again, I don't think that we have those anymore.
Wow.
But the cottonmouths do look so similar when they're young.
It does make me wonder, would there be water snakes, at least in the pond there?
I have never seen any. Yeah, there's supposedly rough green snakes out there, but I also never captured any of those. But they're gonna be a little bit higher.
Yeah, for sure. And then here's something, you may have answered this already. Another viewer says, I have noticed an increase in king snakes, and is this a threat to copperhead populations in small areas? Maybe so.
It could be. I think that they require a larger habitat, kind of like we have barred owls in the forest, but not great horned owls. The last time that Steve saw a king snake out there was probably in the 80s, Dr. Reichling. I did not capture any. The study that was before mine never captured any rat snakes. As they get older, melanate and can look very similar to king snakes. So I just recently did hear a report about a king snake out there, but I think we would need to look at those and just verify. It would be really cool if there were, because that brings another top predator into the ecosystem. And sometimes in those urbanized habitats, what you lose are some of your top predators that are important. And so that would be really great if we did and just increase our biodiversity. But I did not ever catch one.
Yeah, really interesting. Here's another question. In looking for copperheads, did you encounter significant numbers of cottonmouths and or rattlesnake? Oh, you've already answered.
No, when I was hoping to see we have some population of timber rattlesnake in this part of the state. So I was really hoping that when I was out at Neiman that I would see one. I did road cruising, I did traps, I did walking. I never saw a timber rattlesnake. I got a picture of one on a road in between the biological field station and the state park. But snakes in general are declining across the world, which is one of the reasons why we wanted to focus on snakes, is that their populations are declining. So when you talk to some of the older herpers that used to go driving those roads, they would say that they could find snakes pretty regularly, pretty easily. I drove roads and had a very difficult time finding snakes up there. So we definitely didn't find any rattlesnakes. And then the only king snakes that I ever found were in my rural site.
Are there any snake bites that can result in a fatality?
I would say probably the rattlesnakes, and then cottonmouths. They have a different type of venom than copperheads do, and so those are like hemotoxins. So those definitely need to get treated. I would say that with any you receive treatment, because again, with a copperhead, you can have a secondary bite. You might not be sure what it is, so definitely seek medical attention. But I'm pretty sure my dog was bit by a copperhead when I first moved here, and I thought it was poison ivy, and later I realized it was probably a snake bite. And she's 40 pounds and survived it. So, you know, it can really depend also on the amount of venom you receive. You know, animals can give dry bites, but it's really a last resort for the animal. It's energetically expensive to make venom. They don't want to use it on you. They want to use it on something that they'll actually get to eat later. But rattlesnakes and cottonmouths are ones in America that you should be more concerned about.
And then the Carl snake, I guess, yeah, but we don't have that here.
Oh, yeah.
Yeah.
No. And those guys don't look as dangerous as they are.
Yeah. So, and then Beth here says she just wants to say thank you for an excellent presentation, and I will second that. It was just wonderful. I learned so much, and we'll ask you back again sometime. You were just great. So thanks, Maya. Really appreciate it.
Well, thank you guys for inviting me, and I hope that everyone learned something new. I hope that they can go out and appreciate all the native taxa that we have, and not just the pretty cute ones.
Yeah. Well, yes, I really want to get out there and study snakes now. So.
Yeah, if ever you need, you know, a cure for snake fear, if you Google snakes wearing hats, it's pretty adorable.
I will do that right after this. So listen, have a wonderful evening. Thanks to everyone who showed up for this presentation tonight. I really appreciate it. And if you want to watch it again, it is being recorded and it'll be on our YouTube channel shortly. So thank you again, Maya, for your time.
Thank you, Kathy, for your invitation. Y'all have a good night.
Bye-bye.